slides. First off, sometimes they upload screen, but in the meantime, we have 3 trays or blue rack, test rack or test tube racks right here, shown for your unknown on the left side, NA slants in the middle, and utility tubes on the right-hand side. Probably just focus on this side, inverse on your side. But the idea is that each of you are going to select or take one of these tubes, your unknown assignment, and I'm going to ask you once you do to let me know your number, so that way I'll be able to identify your specimen down the road. But once that occurs, you're going to be essentially doing an NA slant or making use of the NA slant to subculture your bacteria. So remember, aseptic technique would be spraying some 70% ethanol to your veg stocks before beginning any process. And then you're going to remember aseptic technique of flaming your alkaline loop, right? Making sure that it's red, so very blue. And the lid of your tubes, both tubes of your unknown and of your NA slants. And then once that occurs, we're going to make zigzags similar to what you see actually in your unknowns. So try to mimic that. Essentially, what's to come is essentially in a week's time, you're going to be making use of these subcultures for future experiments. So the idea is that you want to showcase today successful subculturing technique. And so we'll show it also on this slide shortly. But remember, aseptic technique is key because you don't want any bacterium from the airborne environment to seep into either your unknown or into your NA slants. And we don't want that to occur, so that way you're easily able to identify and characterize your bacteria. Any questions regarding that? Okay. And then once you've done that step, there's also one of the tests that we'll be doing today, a utility test. And so you're going to essentially be using one of these diagrams, essentially, similar to your inoculation loop, adding it about a 1/3 inch deep of bacterium from your unknown, and then plunging it 1/3 deep into the agar itself. And then once that's done, when you're done all your tubes, making sure you label them with your name, practical number, which is 8, and your ID, bacterium, you place them back right here. Now I'll be placing them into the incubators. So that's something to keep in mind. Any questions regarding that? So there are 2 main steps of what we have to do for today. I just want to also share, very important, make sure that you don't stab into your unknowns. So I'm sharing this so that way no one makes into any mistakes wherein they dig too deep. When really for the NA slants, you really just need a small amount, just like what we've done before. But as you can see from the lab manual, you don't have to stab deep inside any of the slants, except for the utility testing. Another question is, okay, touched upon everything. When you're doing subculture, you're starting off from the very bottom, and then you're going all the way up. So you're making the zigzag, also making sure that you're not like stabbing deep inside your name once again. But just remember once again that you're labeling your tubes, so that way I can easily identify them down below. So that is at least a 1/3 of today's practical for you. Any questions regarding this, or even your unknown assignment? Any questions? Does everyone know what their unknown assignment really revolves around? Can anyone give a show of hands as to what they need to do? Or what the goals are? And perhaps what you have to submit next week? Did you raise your hand? Data sheet, that's correct. There's a bit more. So it revolves around this, and we're gonna go back to here, which is the point I talked about earlier. But pretty much, you are making. Some of you have already done this before. Okay, normally I wouldn't show my emails, but the point is, and this is also very important for hazardous waste, to make sure that, please everyone, place them in the right locations. I'll talk about that later on when we clean up today. But the point is the following. Let's say And in case anyone needs my email, I'll just share it here as well. My name, and we just want to make sure that you're all able to see throughout today's lab, the mini labs that we have left. And so it's important that you just pay attention, make sure that you do your best, leave the rest with it. So let's say you have 6 bacteria. Bacteria. So you have 6 bacteria, as you know, the ones that are shown there. And you start off, and your flow chart, I believe it's page 34 of your lab manual. Page 33, I believe, is the table, right, with all the different features, right, from, let's say, the gram stain, the different results, if a gram positive or negative, capsule stain positive or negative. And when you talk about the results and your observations, which is on page 35, I want you to give specific details. What I typically want you to use, rather than just say capsule positive or capsule negative, is in the case of it being positive, encapsulated means positive, and non-encapsulated or no capsule suffices as a negative. But just to make sure that you're very clear, hopefully being able to understand what the different terms mean. Capsule, gram stain, endospores next week. Motility test, we have 2 different types. Does anyone know how we're testing for motility? If they can mention either or. Anyone know? Yes?
No motility testing or...
Okay, that's both of them actually. So essentially, I mentioned it earlier, we have a motility test students with us. And you'll be doing the hanging drop with the depression slides that we did last week. But the idea is that, let's say you have this table, right? It's shown simply like this in your lab manual. Your goal is, for many of you, in case you're not aware, you're going to essentially continue this step or process of identifying and isolating bacterium based on the different traits. Let's say you have 5 right here, 5 bacterium, and you have to specify their names. But in this case, for simplicity, let's say you have 5 here, let's say you have 1 here. And you can kind of tell based on the table, you can artificially already isolate this well in advance. But the idea is that you now are able to separate those 5 bacterium based on the other test results. So let's say it's the capsule stain or endospore stain. So you want to be able to highlight, let's say this is your endospore stain. I'm just trying to show it in different fonts to give you the idea. If it's positive versus negative, you may have 3 here, 2 here. And then the idea is that if you do the motility test or any other test, let's say capsule stain, or their spatial, spatial arrangement, then you hopefully should be able to separate them more and more until you get all of them individually separated. This is important for diagnostics as well. To think about the implications of these results. Let's say you're in a clinical environment and you're a micropathobiologist and you want to see and differentiate different bacteria based on what you get from the OR or from histology readings. This is where a lot of this is actually very applicable and has benefits for you being able to understand the effects of the not just the disease, but the most effective treatment for those or that specific bacterial species. Any questions regarding that? No? Okay. And so if I go back, this is what I was referring to earlier. I'm going to turn on the gas. But let's just make this full screen. So yeah, and this is reiterated here. So just please bear in mind, you start at the very bottom of your nutrient agar plan. I want to keep one in case any of you need extra, just in case. The point is that you really are taking a small, you would say pretty much a little, once again, what you have from your unknowns. I would say go to the bottom, but don't go out inside, or otherwise you go midway in. And then you just essentially spread it as if your bacteria are skating around your agar, starting from the very bottom, going up to the top. That's pretty much what I want you to complete now in the next 10-ish minutes. Any questions? Any questions? Actually, I'm going to turn on the tap. Just take the chair. Question. One. How Has anybody solved the period of years? I think you guys should be okay, but just let me know once you get your hues, the IDs of your bacterium, so that way I can write them off.
Sorry, so we're not all three to three.
You can take the monthly test tubes as well. Is that your question?
Paragraph is 3 2. It's not for this.
Oh. So, okay, when it comes to, so you've probably taken all three test tubes, right? Your unknown, NA, and motility test tubes. This is a good point. The motility test tube doesn't fit in the red rack. What you can do is simply, before you actually do that in your bacterium, place them onto your bench top. Just make sure you spray 70% ethanol. As well, if you wish, you can place them planted onto your white tree. I just know that there's only so many trees that we have, but nonetheless, you can make use of rest in the I'm gonna have to see for give me one. Anyone having any difficulty starting on their Bunsen burners? Before I do, Tara, can I have I was about to ask.
Yeah.
That's but you did this, right?
I did the day. I did it. Okay, so basically, I was gonna do it right here,
That's okay.
but then it wasn't gonna fit, so I did it.
If I were you, I would recommend stapling.
Yeah.
Perfect.
Okay.
Thank you. Everyone.
They're like,
On that. The loop should be on the side. Yeah. Make sure your Bunsen burner is not vaporizing such that the heat can be seen quite readily with the red flame. Someone's Bunsen burner is on too high. Some of your bacteria a bit more viscous and it's a bit more, or some of them are more sturdy. So I understand that's why it's hard to discriminate how much you actually take out. But usually it's just about, I would say, a quarter a match of what your loop looks like. Yes. And just so everyone's clear, you're taking it from near the bottom of your unknown. Like you could be halfway to near the very bottom, but just don't gouge from your unknown, like deep inside. The reason I say towards the bottom is such that you're able to get and maximize as much bacterium within that quarter loop. So that way it's not too difficult for you to keep digging and scooping out bacteria. Just remember, everyone, to make sure you're practicing the best technique to perform, such that you're removing and reducing any risk of contamination. Because your bacterium is what you want to identify, not any airborne bacterium or not anyone else's unknown, right? So just bear that in mind. I can help if needed, but let's see if we could finish it by 35. At least this component of taking out the NA. That's it. That's good. You're going for the motility test tube once they're deep. You don't have to do it 3 times, just one stop to be enough. It's a small tube. That's why the motility test is done in small tube. Everyone doing well? Okay. Yes, anyone can take these daggers or sorry, essentially these rods to be able to poke into their we're gonna put in the if you're done, you're done. That's your sorry. That should be. I'll show you. The big one, just two medium. When you're done, you can place your electrons right here.
You guys return there? Yeah.
Who had test tube 481? Anyone have 481? No one? I hope everyone has at least recorded or knows their number.
Sure. Yeah. That's sure I'm gonna be.
Yeah, but.
The middle.
Yeah.
I got ingredients.
Okay, I guess three.
Yeah, yeah. Yeah, but I'm good with my initial yes.
Yeah, Brian's name, right?
Now, everyone, I'm going to start asking the numbers for your bacteria. So now, in terms of timing, I also need us to actually catch up a bit ahead in terms of timing because we have a few stains that we need to go forward with. I see some people have done the capsule stain already, and some of you look like you're preparing for the gram stain, which is good. But in terms of timing, please make sure I'm going to start asking, or actually, I'll move through a few other things. Someone had a better question. Yes, once you're done placing the tubes, once you're done with the tubes, the three tubes, at least for the NA slant and the motility test tube. Please place them back in the middle and right rack respectively. The unknown tube that you had earlier, you're still going to use it for our gram stain and capsule staining today. So you can still make use of it. Just make sure that you don't forget your numbers. Make sure that you write it down. Yes. Thank you so much.
Yeah.
So someone left a first class. This is
For different stages, you want to learn the same for now. You want to pass it to the
someone left the first on the ground. I'm gonna now turn the slides. Hopefully everyone has at least attempted and is in process of completing their tubes for the motility test and for NA. Please make sure that once you're done to place them in the respective locations where you have them all labeled with your name or initials and the ID and ideally the practical number. But if you've already placed it in, that's okay. Now, in terms of unknowns, I talked about this gram stain. This is the next exercise for us, and you've probably seen this from your lab manual. If you're already in process of doing the gram stain or the capsule stain, that's okay. Continue as you are. But I just want to make sure we're able to complete the informational component of today's lab. And so, for those of you wondering, what's a gram stain? What does it aim to show in your bacteria? Yes. Maintain, yeah, I heard just if you can The primary stain. Perfect. Okay, but now, and that's a great answer, but there's one more thing. What feature discriminates or differentiates between a gram-positive and gram-negative cell? You're close. Pepto something. What's pepto? Glycan. Peptoglycan. You had something else? Yes. Perfect. So being able to understand that, especially when you look back into the past for your lab exam, that you want to see and discriminate, okay, what's the difference between gram-positive and negative cells? The difference of the thickness of their peptoglycan layer is important. And in fact, resonates with why we're doing gram stains and why you see different colors for different statuses of bacterium. If they're positive, crystal violet, which is your primary stain, is going to be retained following the counter stain and following decolorizing with 95% ethanol. So bear that in mind. Everyone should have those small cubicles with 95% ethanol with a silver glass tray. That canister is for your ethanol. Bear in mind that it's not too close to the Bunsen burner.
Guys, can you move the ethanol?
So just make sure, everyone, you're paying attention. That when you look now into the other components of today's lab, you want to be able to see within your slides. You only need 3 slides for today at max. For your unknown, for your positive and negative controls. And the negative and positive controls are in your lab manual, and the bacterium are found in your trays. So you use them as groups to be able to isolate and do perfect smears on single slides. Please bear in mind that you're using single slides for this exercise. Use essentially, I'm just going to skip to this slide, slide number 6 as shown here. Make sure that you have on a single slide all 3 smears, your negative, your unknown, your positive, right? As shown in the lab manual. And make sure that you have them labeled so that way you know on the left side is your negative or positive. In the middle is your unknown, and the positive or otherwise negative, essentially here positive, is on your right side. Just make sure that you're able to also make a note of your initials, the practical as well, because we want to make sure that you have been accurately representing your slides, and that way it will be easy to identify. Yes. Okay. Who has, have you checked your bench? Okay. I'll check. Make sure once again your NA slants are not being placed in the unknown rack. They're supposed to be placed in the middle rack, please. Just bear in mind that you're following instructions, so that way you'll be able to ace the class. Yes. First off, I'll ask people their numbers and we'll find them. But you've already done the motility test, right? And the NA slants, right? Actually, check the middle row, just in case you place it inside there. Everyone, I'm just gonna start asking. Hopefully everyone has done their motility test, the NA slants as well. When you're done, I need to make sure that there are 24 tubes in those 2 racks. So please let me know when you're done. If you make any spills, let me know as well. If you have stains on yourselves, I would say continue, or we actually have lotion on the right side of the emergency sink. So just make sure that you have it. Okay. Does anyone have test tube 374? Anyone have test tube 374? It's an unknown, but it appears that someone may have used another person's unknown, which we don't want happening. But I just want to make sure. Who has test tube 374? Maybe took the wrong unknown. Okay.
Each 3 drops of water. Right?
Okay. Technically, both of you can use it. I know you're finishing up, or are you done? Perfect. Okay, so now you have to go share it for your other tests. I have to think about this a bit more now. You did, both of you done multiple tests. Let me just see if there's any more. Okay, in the meantime, make use of it. I'm gonna start asking people their test tube numbers and see if there's any additional ones. We can't do this. I'll find another test tube. So don't worry. In the meantime, continue as you are. It's okay if, like, for my marking purposes, it's okay, but you guys may be stuck in timing because you need to technically share the same one. Otherwise, like for the other things. So what I will say is, you can also make use of your subculture. Until then, I'm gonna ask everyone their numbers. If I can find an extra one. That's okay, but I at the same time, this is a hefty lab, so I want to make sure I may be able to find one for you guys. In the meantime, stay seated. If you want to prepare yourself slides, that's perfect, even with the negative quadrant. I'll start asking people their numbers. Just remind me yours as well. Thank you. Distilled water, and okay, I'll do it. In the meantime, mortality test tube. Okay, I have to ask you that. Remind me one more time. Or give me some time. Thank you so much. So, yeah, I'll do it for you. Before we move further, I just want to make sure I have everyone's IDs. So this is the perfect time for you to tell me. For Lila or Lila, can I have your ID? What's your unknown number? Lila? Lilaa. Okay. Hajra, can I have your, sorry? 385. Zara, what's your unknown number? Sorry? 394. Amro? 395. Cynthia? 405. Carlos? Jacob? What's your number, Jacob? Yeah. 882. Melina or Melina? 375. Shauna or 384. Alta? Yes. 474. Yes. Yes. Make sure you're using your right unknown. Make sure you use yours and not someone else's, please. Now in terms of, okay, I'll continue. Ravati, your unknown number 485. Vice Navi 44. Sahar 391. Taurus or Charis? 374. Okay. Yes. Yes. The slide from the microscope. Okay. Quick question. Did you do the gram stain or okay, I'm gonna come to you. Once I have two other students, but I'm probably gonna go to you first. Can I get numbers? I'm gonna try to ask everyone's numbers. You have? Oh yes, you have. Okay. Funmi or Funmi? 373. Zabeen? What's your number, Zabeen? I'm sorry if I'm saying anyone's name incorrectly. Forgive me. I'm not trying to do it in any way, shape, or form. 1313. Okay. Ashwin? 42. Garvin? Garva? 392. Saad, 374, right? Yes. Okay. Now Scott, 103. Zat, yes. 885. 881. Terry Lou. Terry. 163. Fatima. What's your number? Fatima. Qamar? Yes. $471. And, sorry. Okay, 481. Okay.
On one slide? Okay. Yes. We go with our castle. I'm going to. Yes, the capsule is very a positive and I'm pretty sorry. I just
Okay, yours looks okay. The middle one looks the middle one too fine. A little second. Give it like a minute or two.
Well, that one you're supposed to do, or like
What's the positive?
lactis. No, two different slide. Yeah, but he's asking bacteria on the one side, right?
One human, one bacterial.
That lactis.
Whatever.
Yeah.
Yes, you mind?
The capsules in there, one hour. It's like, oh, you want to do this?
Yeah. No,
Have you already done that? Yeah. Okay. You should try that. Yeah. So, one question for you. You should not do it. I have spoken about that. Why is it water don't put any liquid out, have to be sterilized. We did the whole thing, right? But
you're all on the same page with a few things. I'm very happy that many of you are catching up and are ahead of the game. But I just want to make sure that the basics and fundamentals are also fulfilled for all of you. Please make sure you're following aseptic technique and you're being mindful of the chemicals and materials that are around your Bunsen burner. Very important, including yourself, that you don't get too close with your lab coats, but also make sure that ethanol, especially 95% ethanol, is not too close as well. Please bear in mind, because for safety concerns, we don't want anyone getting flamed up, and we don't want to also use the shower to cool you down. So bear that in mind. Before I answer following questions, let's go straight to the gram stain, or sorry, the capsule stain. May I ask if everyone's done the gram stain or almost done? Raise your hand if you're now in the capsule stain or already done that, which is also good. But like either or, if you're in the process of doing your capsule stain, raise your hand. Okay, I'm going to start asking by groups where you are in terms of exercises. Exercise 1 was inoculating your subcultures. Exercise 2 is your gram stain. Exercise 3 is capsule. So let's start asking questions. Amro, what's your group looking like? Where are you guys at right now? Number 2, gram stain. Okay. What are you guys on right now? Gram stain? Gram stain? Are you guys on gram stain or capsule stain? Gram stain? Okay, gram stain. Okay, and a mix of two? Okay, okay. Okay, so I'll wait then before moving further. But please, everyone, bear in mind that you're being not just proficient, but mindful with what you're doing. Please make sure that you're using the right unknown. You're not using someone else's, because otherwise you're going to get different results. right? Because you're comparing the negative and positive controls for gram staining and capsule staining with your unknowns. So just bear that in mind, everyone. And if you need help or support, just also let me know. Thank you. Um, just before I do, sorry, we have the capsule. Um, Anyone using this slide, just in case, I know some people are going to the capsule stain. I'll move forward to the capsule stain if needed. But the point is, pretty much after the capsule stain, motility test, acid fast stain, and that's pretty much like we're ahead. And I just want to make sure, I don't want anyone getting stressed out, please. You don't have to get stressed out. Let me know if things are a bit too quick and we could slow down if needed. But I just want to make sure that you're able to understand the concepts and what I've touched upon thus far. And slowly we'll talk about the capsule stain, likely in the next 15-20 minutes. I want to make sure you guys can complete the gram stain successfully, as it will be great prep for your unknown lab assignment when I mark your slides. So bear that in mind, everyone. Make sure that you've labeled your slides as well with your name or initials, the unknown, and the locations of your positive and negative controls. Yes, Ashwin. Okay. On five, anyone want to please.
Here.
What do you need? Always have a clear bottle to be able to make use of. If you see a decolorizer on your bench top, that's what you're going to be using with your crystal violet to essentially complete the gram staining technique. Yes. Manual, but I'll show the slides. This is simplified. I will go with how it's shown and provided in the manual. But now we're counting down, so I would just recommend keeping an eye on time. We're about 15 seconds in, 15 seconds. And then when you need to use the decolorizer,
Everyone's 40 hours.
Yeah. Yeah.
I do different, I Georgia.
Everyone's following. Is anyone, here for example, safety purposes?
Oh, you color it?
Okay.
Yeah?
It's okay. Like how much is that? One is okay. One have to.
Okay.
One would be.
I do.
Right. Yes, yeah, no. How do you do that? Is it also Ramsey?
Right.
Check to see that it's like actually dry. So
I don't tell you, I did it last week. That's not. He said. He didn't answer.
Yeah.
All of them. No, I'm saying, yeah. Yeah. So, yeah.
How do we figure it?
That's like. The air dryer, that's just how air dryer. Yeah. Yes. Yeah. There's one here.
Oh. Do you need any more?
No, I thought.
Someone's slow. Someone's playing is on too high. I think you could complete a lot of yeah. In between the two objectives, lenses, holding.
Put it there and
Yeah.
I still don't understand what you were saying before, why it would be positive if it's supposed to be.
So that's where. It's good that you're this.
Say it again. No, you just put it on the red combo and black combo, right? Yeah, both of them, red combo, you're doing the same things for both. I put red combo on it. Yeah, because you're repeating it, you're doing the same thing. Like you do red combo for both, you're essentially doing the same thing for both. Okay. So red combo will be out, and then red combo. Yeah. But I don't. Objective. Well, there.
Yeah.
Yeah. But.
How are you doing so far? Are you on the capsule stage, ground stage? Okay. How many of you are on the ground stage? Raise your hand. How many of you are on the capsule stage? Raise your hand. Or have completed one stage technique already? Okay. Have any of you from this side completed one stage already or no? Any? Anyone? You can see one.
Yeah.
Okay. Anyone else completed one stage on this one stage here? See if you guys can get another one. Yeah.
There's supposed to be some red within purple. Yeah. I don't know what time to. Did it? But maybe it was around one. Right.
Depending on time, they have to use a microscope and have shown me their experience, we can start using another set of microscopes. Especially those behind me at the back right side. I apologize that the microscope aren't working too well, but you can make use of the other adjacent particles. But make use of pattern. No, we're getting on.
Yeah. No. Thank you so much.
Carlos, do you want to plain slide? Capsule?
Yes, please. Oh. Excel set it up.
Everyone done the ground state, to confirm, or nearly done? You can draw them in your lab notebook. You can take pictures as well. Make sure that you're able to make a little shape, the color, the little. Okay. See, before you do, just before you do, I'm gonna ask, or sorry, share some updates and common questions that will help you out, all of you. So, how's everyone doing? Yeah, I know it's a bit tense, but alhamdulillah, I'm very happy that you guys are like completing multiple stains at once and pretty much are concluding today's practical. So congrats and very happy on your performances. Forgive me if I'm also a bit tight on time, but what I'll say is the following. Many of you have done your smears and stains, and pretty much one of the common concerns is that you don't see just a clear purple or pink for your unknown, but sometimes see a mix of both. What is that called? Has anyone seen it from the lab manual? It's related to this to a degree, but there's another term, gram something. I'll talk about it right now. Gram variable. So the idea is the following. And you could probably also see this in your lab manual if it's like you could see it online for color. But the idea is that you see a mix of both. And in practice, in theory, your bacteria should only be one or the other, right? But sometimes when you're visualizing, you are seeing both. And that doesn't mean that your bacteria are both, directly gram positive and negative. Like they wouldn't typically have both types of kind of like layers, thicknesses. But the idea is that you've likely over-smeared or under-smeared your bacterium. So bear this in mind. Everyone, I just want to make sure you're paying attention because there's a few common concerns that I've noticed for many of you. It's your smearing technique and your staining technique. Smearing, you don't want to do too much, which is often the case. If you do too much, there's so many layers that it makes it more easy for you to miss out on staining appropriately. And that's where you often get gram variability or a mix of both appearing types of bacterium, even for your single species. So that's one common concern. It's a limitation that you can talk about for your post-lab. The other is your staining. So timing for the slides that you saw before, it's very important that you match and accordingly stay accurate and true to the different chemicals and liquids that you're using. So for example, crystal violet as your primary stain, 1 minute is how long we're using it. If you overdo it, if for whatever reason, before you do your next washing step, you've kept it on much longer than a minute, right? Before actually getting ready to begin washing off crystal violet, that adds on extra time, meaning that you're likely gonna have false positives, especially for your unknown or especially your negative control. So bear that in mind, because I know sometimes you may get just your purple across all different layers or all different conditions, but typically that's just by slide view. If you have a closer look, you may also be able to see pink bacterium, which can indicate your gram-negative conditions. But just bear that in mind as one common trait. It could also be the alcohol, depending on if you use 75% or 95% accordingly for decolorizing. And then safranin if you use it for the appropriate amount of time. Also, make sure that you heat fix, right? Which is different from the capsule stain, but it's a separate point that can be noted. Any questions regarding that? Yes. Yeah. And the literature is pretty much, great question. The literature is pretty much what you see from your table, page 33 of your lab manual. So you should be able to see what the observations would appear like, the arrangement, the size, the color, or essentially the result of your tests. So bear that in mind. You can grab your phone. No problem. So I just want you to all bear that in mind because for the assignments, you have to submit to me pages 34 and 35. 34 is this one. 35 is the one with your observations where you have to tally in the different points on size, the different categories that are mentioned for the results. Some of which will come up next week, so you don't have to worry about all of them. But apart from that, your assignment is pretty much these two plus your slide. The slides I'm going to talk about in due time. I just want to make sure that you're able to do at least the capsule stain now. So I'll give you guys about, I think I gave actually more time than needed before. So I hope about 5 minutes should be okay. Because many of you have already passed that step. Like some of you went first to the capsule stain, then to the gram stain. And I know I said earlier that we'd have 15 minutes, which would have been 10:30, but we're almost 40 minutes ahead for past time. So likely you guys already went to the capsule stain. This one, just giving it a few more minutes. Well, for time, how about to make it easier, 11:15, 8 more minutes. If you haven't done, if you're finalizing observations for your gram stain, you can. For your capsule, you can as well. Once you're done, also let me know. Just make sure you label your slides accordingly. And then the motility test. Hopefully everyone has done the motility test. Right? Missing two more things here. Unknown, not yet. Yes, but now you think of your absolutely.
Yeah.
Who's got? Anyone else? This, but you're also keeping in mind. Okay. I didn't also answer your question. Sorry. Your question one.
One for the negative. I need to get pictures of young. Okay, so I should get those.
Yeah, so we did really positive and negative.
I haven't done the capsule thing yet. Just did that.
So look at the agency positive and negative. Yeah.
Oh, I love to do this.
Yeah. In here, because they were using our iPhone.
So okay. So okay. If you've done, so my previous practical has done capsule staining for the unknowns as well. It's good for observations. If you've only done it for the positive, that's also okay. Like I won't, let me just see the pages because I don't want you also to lose marks. Do in the meantime, we still have time, so try to do your unknown if you can. It also only takes 5 minutes, pretty much. So if you can do your unknown, that's good. It's good for your observations as well. Sorry, any other questions? What's everyone doing so far? If you can do the capsule stain, that would be amazing. If you are behind on your gram stain, then continue preparing your slides. It's okay. Question: hanging job we're gonna do as like yes. If you are ahead, then you prepare your hanging job. Okay. That's okay. Okay.
I was just doing brand and solution.
Program, capsules, utility. We had the hanging job as well, and four. Oh, there's five. And then like the NA stuff.
Okay, because I've already used the brand. I need some good stuff.
Did you view them?
No, I don't do that.
Okay, have a view of them. And see what you can do
You know when you take this
for.
for the NA,
You can wait on that.
it won't get smeared off.
No.
What is it, the tube after?
Two, you place it in the middle string. Nope.
It has a picture I took from my unknown,
Okay.
and then I went back to the gram stain, and it came up as this.
That's what's your unknown? Or
Yeah, also mine.
What's canine is your beakers. Those you see those yellowish beakers right behind that microscope?
Do you think maybe dye from the other side, like the oil version, like stuff?
I doubt it. It could be just another location within your smear that has this color.
This color, okay, should I trust the pink then?
How can I have a look at it?
Or see?
And sorry, you had a question. Okay. Oh, both of them are saying that. I got this. Yeah. Okay, maybe to answer, you have both of you similar questions. This is my recommendation. If anyone's seeing variable results, that could indicate gram variability, but also it could depict how much of a smear you have that is gram negative, and the other part that could be gram positive. If you see a more bulky part or a more greater proportion of it being gram negative, then that indicates for you that your sample is more likely unknown gram negative. If you see the opposite, that more of it is gram positive, then assume that it is gram positive. But we're gonna repeat this process soon, or like in future weeks, maybe next week. But the point is, see if you can complete it for now, and then I could we'll likely talk about it soon as well afterwards. But don't worry, I would say really try to compare and differentiate your unknowns from your controls. That'll make it more easier to determine. Is that okay? Yeah. No. Oh yeah. We're done.
Do you want your, yeah, you're ready.
What's yeah. You've already done the Matilda test, right? The one in the yeah. Yeah, so the one that we the one on the third side, like can I show you? Okay, so. You can also, you can see the graph. Then that's. And you can observe
And yeah, you bring stuff.
Yeah.
After you rinse the stain off the water.
Also, issues we can't. Yes. You just, that should be okay. Like, we see this is good. You can have these are likely for you. So to be able to see what have you done? You don't need
Yeah.
If your group or yourself are done both the capsule and gram stain, you can make use of completing exercise 5, which is pretty much a demo, acid fast stain. Just have a look with your group at one of the slides and just compare the different colors that you see. Some are blue, some are red. Just be able to know which one you see. And then return. Thank you very much. I was just wondering if you've done Let's try.
Oh, I'm fine. Yeah, I'm not. I think. Oh, he's smart. Yeah, he's smart. He's smart. Yeah.
It wasn't about. Yeah.